Good morning, students. Today is our eighth session in microbiology. Today we will speak about bacterial nutrition and how to provide the nutritional requirements for the bacteria in the laboratory using prepared media. First, we are going to speak about bacterial nutrition and the different types of groups of bacteria according to their nutritional requirements. Bacteria always differ in their nutritional requirements according to their habitat where they live. That is why they have different nutritional requirements. But these nutritional requirements are grouped into three main requirements, which is the energy. For the bacteria to get nutrition, it requires energy, as we explained in metabolism, catabolism, and anabolism. So, energy is an important component for nutrition for bacteria to synthesize its food. The other uh, category, which is very important as the nutritional requirement, is the electron donors. And as you know, to prepare any food in the cell, which is a metabolic process, there is two types of processes: oxidation and reduction of chemical processes that takes place in the cell for for biosynthesis or anabolism and catabolism. So wherever there is an oxidation process, there is in the same process there is a reduction process. So when we say electron donors, that means these electron donors are part of the oxidation process, so giving electrons and receiving positive charge or anions. So this is why we call most important for any building or biosynthesis of nutritional products, there should be electron donors, which is opposite to oxidation process. The other category, which is very important for as a food as a nutritional requirement, is the source of carbon. So there are three important requirements for a bacteria, which is constitutes the nutritional requirements for every bacterial cell, which is energy source, electron donor source, and carbon source. They differ in how they get these three requirements. Either is it an organic compound or organic compounds. So based on the source of energy, we have the first group, which is called phototrophs. These the bacteria, and we explained before that some types of the bacterial cells, they have photosynthetic dyes or chlorophyll. And some other they have chlorophyll like dyes in their cells. So phototrophs, they get their energy from radiant energy. That means from the solar sun, from the solar energy. So these bacteria, they have in their cell the pigments, which are chlorophyll and other types of pigments, which help these cells to capture and get use of the solar energy. That is mean the radiant energy or the sun or the light that comes from the sun. So they use this light to produce ATP as a source of energy for the cell for building their nutritional needs. Such bacteria is example the cyanobacteria, the sulfur, green sulfur bacteria, purple sulfur bacteria, and purple non-sulfur bacteria. So they differ in the type of pigment, but at the end they have photosynthetic apparatus and they have photosynthetic pigments. The other type of bacteria is the chemotrophs. So these are the bacteria that obtain their energy from its name from chemical reactions. So these chemical reactions, it could be definitely it is an oxidation. So there is on the other side in the same reaction there is reduction. But here in chemotrophs we are speaking about oxidation. So this type bacteria it oxidizes chemical compounds to get its energy stored in the form of ATP. They differ in the type of of compounds, chemical compounds, either inorganic or organic. So those bacteria which oxidize inorganic compounds to obtain energy, like the compounds like ammonia, reduced sulfur compounds, ferrous salts, and molecular hydrogen. So there are different types of bacteria. Each type of bacteria use one of these inorganic compounds. Example like cyanobacillus, it oxidizes sulfur compounds. Okay, so if it is oxidizing sulfur compounds, that means there is an oxidation reduction process. So it oxidizes sulfur compounds to produce energy. Nitrifying bacteria, it oxidizes ammonia because ammonia it has a nitrogen component or nitrogen element. So it oxidizes ammonia, please, into ammonium compounds to produce the required energy for the cell. The other type, for example, the iron bacteria, it oxidizes ferrous ions into ferric ions. So that means it is producing energy out of this reaction to be stored for the cell for any nutritional requirements. Other type of bacteria, it uses molecular hydrogen. It oxidizes molecular hydrogen to obtain energy. So all these types of bacteria, they use inorganic compounds. Another category of bacteria, they oxidize organic compounds. That means compounds that contain carbon and hydrogen in their organic shape. So this includes actually this group of bacteria. Most of the bacteria are found in this group. They oxidize organic compounds to produce energy. Example, Escherichia coli, Bacillus, and other type of bacteria that oxidize organic compounds, organic carbon compounds, so to produce energy and store it in the cell for synthesis of the nutritional um, requirements. So this is based on the source of energy. The other category, or according to the other requirement for nutrition, bacterial nutrition, is dividing or grouping the bacteria according based on the source of electron donor. As I mentioned before, whenever there is an oxidation process, chemical process, definitely in the same process there is a reduction process. So according to the electron donor, so there is one group called lithotrophs. These are bacteria that utilize inorganic substances, the same inorganic substances that we mentioned based on the source of energy, which is sulfur compounds, ferrous salts, ammonia, ammonium compounds, and molecular hydrogen. So they reduce these compounds, okay, and at the same time there is an oxidation process. So lysotrophs, it's equivalent to what we call chemotrophs. There is an oxidation process, then there is reduction process. So they utilize these compounds as donors, electron donors. So this is what we call a reducing power for biosynthesis. You need to reduce, like in the catabolism, you need to reduce certain compounds so that you have minerals or elements that can be oxidize further to produce energy. So it's a reduction oxidation process. This group that uses the inorganic substances as electron donors, we call them lysotrophs. While those that use organic organic compounds, we call them organotrophs, as indicated by its name, organotrophs. They use the organic compounds as electron donors. So organotrophs, they use organic compounds, unlike lithotrophs, they use inorganic compounds. The third type of, according to the requirements, nutritional requirements, is the division or grouping of the bacteria according to the base on the source of carbon. The first group, we have the autotrophs. Autotrophs, these are bacteria which use inorganic carbon compounds like carbon dioxide. Okay, carbon dioxide is a source of carbon, but is yet it is an inorganic compound. So they have the ability to fix carbon dioxide and reduce it, because you need to reduce it into carbon. So it's a reduction oxidation process to use this carbon into organic matter. That is synthesis of their nutritional requirements from organic compounds. The other type of bacteria or group that are grouped according to the source of carbon is what we call heterotrophs. So these bacteria, they obtain their carbon source from organic substances like methane, paraffin, and other types of organic molecules according to the type of bacteria and the environment or habitat where it lives. So the majority of the bacterial genera are part of these heterotrophs. In some types of what we call autotrophic bacteria or autotrophs that utilizes inorganic elements like carbon dioxide as source of carbon, they utilize carbon dioxide and organic organic compounds together. So this type of autotrophs, we call them facultative autotrophs because they can utilize both carbon dioxide and organic compounds as source of carbon. As you can see, we grouped the types of bacteria according to the requirement, according to the nutritional requirements. But definitely, as I mentioned before, all these three requirements are required for each bacterial cell. That is why they are grouped and named. The bacteria is named based on the grouping of the three categories. So we have the nutritional subgroups of bacteria, and this is how you name a bacteria according to its nutritional requirements, including the three components that is needed for nutrition. So for phototrophs, they can be photolysotrophs, and these utilize light as source of energy. They use inorganic compounds as donors of electrons, and they use carbon dioxide or organic compounds as source of carbon. Photoautotrophs, it's for organotrophs. They use light. They use organic compounds as electron donors and organic compounds also as a carbon source. So whenever there is and I mentioned before, there is an oxidation process. In the same process, there is a reduction process. Photoautotrophs, they use light because they are all photo, so they use light as a source of energy, inorganic or organic as electron donors, and carbon dioxide, which is an inorganic compound, as carbon source. Photo heterotrophs, they use light, they use organic as source of electrons, and organic also as source of carbon. Chemo, when you say chemo, that means they don't use lights, they use chemical compounds. So they differ in the type of chemical compounds. So for source of energy, they use chemical compounds. For electron donors, they use inorganic, and carbon dioxide or organic for carbon source. So these are chemolithotrophs. Chemorganotrophs, they again chemical reactions for energy source, organic for electron donor, and organic for carbon source. Chemoautotrophs, chemical as a source, chemical reactions for source of energy, inorganic or organic for as a source of electron, and carbon dioxide, which is an inorganic compound, as a source of carbon. Chemoheterotrophs, chemical compounds for as a source of energy, organic and organic compounds as a source of electrons and carbon source. So from the diagram below, you can see a single cell. It has the three nutritional requirements. They are integrated together. They have to be working together in a single cell to produce macromolecules. For energy, as you can see, for energy source, it provides ATP, regardless of the is it organic or inorganic compounds or is it light. Okay, at the end product is ATP. So ATP is required for all the processes that is leading to the production of macromolecules. When we say macromolecules, this is a nutritional component that is required by the cell. It could be a protein, it could be amino acid, it could be um, lipids, whatever the macromolecules are. This reaction, again, it requires carbon source. Of course, the carbon source can be organic or inorganic. At the end, it is a carbon source. It takes part into the process of producing a macromolecule. This reaction, for the reaction to happen, oxidation reduction, there should be an electron donor source. So the electron takes part in the oxidation process, in breaking down the molecules into small molecules for biosynthesis or building the products that is needed for the bacterial cell. So the three components of the nutrition that is required by the cell, they integrate with each other in one process to produce the macromolecules that is used by the bacterial cell. So if we want to define each bacteria according to its nutritional requirements, for example, when we are going to define this according to their this is how you define the bacteria. So for photolithotrophs, these are bacteria which obtain energy from light but use inorganic substance as electron donors. For example, they use hydrogen sulfide. They oxidize hydrogen sulfide into its sulfur element, and of course, because it's an oxidation process, then it produces electrons. This electron takes part of the reaction that is building the biosynthesis of macromolecules. So, as you can see, there is an chemical oxidation reduction chemical reaction. We call it photolithotrophs. Photo because of light, litho because of inorganic compounds, and this type of bacteria is green sulfur bacteria and purple sulfur bacteria. So of course these are the type of bacteria that uses sulfur as its as its inorganic compounds. Of course, lysotrophs, photolithotrophs, they call all other types, they use other inorganic compounds like ammonia, like nitrifying bacteria. Again, it is called photolithotrophs since it uses inorganic compounds for as electron donors. So the second type of bacteria is the photoorganotrophs, and this, as its name indicates, it uses light as a source of energy, but it uses an organic compounds for as electron donors. So this type of bacteria is they uses organic compounds like pyruvate, succinate. For example, succinate it is oxidized into fumarate and it releases electrons from this reaction, and this is a source of the electron donor for the reaction for the building of the macromolecules in the bacterial cell. This type of bacteria is like that. An example of this bacteria is a purple non-sulfur, those that have pigments but they are not able to use the sulfur as an inorganic source of electron donors, so they use organic compounds like pyruvate, succinate, formate as electron donors, as indicated in the chemical reaction. Photoautotrophs, these are the phototrophic bacteria which obtain their energy from light, but at the same time, they use carbon dioxide as a source of energy and as a source of electrons. So it's an oxidation of carbon dioxide, source of energy, source of electron, and source of carbon. The other type, the fourth type, is the photoheterotrophs, and this is the bacteria. They obtain energy again from light because they are photo, and carbon from organic substances. Of course, there is a reduction in and and the oxidation reaction. Then there is a release of electrons, and there is a reduction of organic compounds into carbon as a carbon source. So the third type is the other type. Then the other group is the chemolysotrophs, and this is a bacteria that use the same inorganic chemical substances as a source of energy and and electron donor. So they use inorganic since they are called lyso. So any bacteria that is called lyso, that means it's using inorganic compounds. Organos, that means it's using organic compounds. And the more and always lyso, they use inorganic compounds as the donors of electrons and at the same time as a source of carbon, if they are using carbon dioxide, of course. But there are others that use other type of chemicals like sulfur, like sulfur compounds like ammonia and so on, so they have to use other source of carbon like carbon dioxide. Chemoautotrophs, these are the bacteria which obtain energy from oxidation of inorganic compounds or carbon from carbon dioxide, and they are close to chemolysotrophs. Chemoheterotrophs, these are the bacteria which obtain their energy from oxidation of organic compounds and carbon from organic compounds. So they use as organic compounds as a source of electron and a source of energy and as a source of carbon. So that's why we call them chemoheterotrophs. The other part of this session is how we culture or we grow the bacteria in the laboratory. Of course, to grow the bacteria in the laboratory, you need to provide all the nutritional requirements for this bacteria and all the conditions, the pH, the temperature, the oxygen, and the osmotic pressure that we already requirements that we spoke about before in the previous lectures. You need to provide this for the bacteria to grow in the laboratory. That is requirements that fit its normal habitat, the normal environment that usually grows in. So, what is bacterial culture? Bacterial culture is a method that allows the multiplication of the bacteria on a laboratory medium, providing all the conditions that is required for its growth. So this is what we call bacterial culture. So it is a method of growing bacteria in the laboratory on a medium. A medium is the environment or the habitat that the bacteria is able to grow and multiply and increase in number. So these conditions, of course, the nutritional requirements, as we saw in the different types of bacteria, we already spoke about. If it is it's a chemolithotroph, or is it photolithotroph, or is it a chemoorganotroph? That means each type of bacteria is having its own requirements, nutritional requirements. You need to provide these requirements for the bacteria in the medium so that it can utilize these requirements and grow and multiply in number. So, what is the advantage of growing this bacteria in the laboratory, or culturing this bacteria in laboratory? The first thing, if you want to identify and know the morphology of the bacteria, you need to culture it. So, the most important thing is to identify the morphology and the characteristics of a bacterial cell. So, you need to grow it in the laboratory for further investigation. The other important reason for culturing bacteria is for diagnosing infectious diseases, as in the case of the medical laboratories. So, how do they diagnose these diseases? They take sample from the patient and culture it on a medium in the laboratory, and then they will be able to know which type of bacteria is affecting the patient so that they can give the right antibiotic. So it is important for diagnosing infectious diseases. The third important reason for culturing bacteria in the laboratory is the production of vaccines. Of course, if you know the cause of the disease, then you can provide a vaccine to combat or to protect or to immunize people against diseases. For example, the vaccine of tetanus, the vaccine of diphtheria, the vaccine of Bordetella, which is what we call in Arabic shahook, that gives these very severe cough. So all these are bacterial types. Very few of the bacterial diseases that have vaccines. There is a vaccine that we usually use in Sudan, for example, the meningococcus bacteria, which is called the Suhai. So all these are vaccines developed from the causative agent of the bacterial cells that cause the disease. They are grown in cultured in the lab, and then they are processed to with different methods for production of vaccines, or what we call serological antigens. Okay, so other reasons why we culture bacteria in the lab is the is for genetic studies. For example, if you want to know the chromosome structure, the content of the genes of this bacteria, as in now in the science of molecular biology and genomics, they study the genetic content of this bacteria. Why? Because they want to utilize this bacteria for the benefit of the human beings, either producing genetic manipulated vaccines, or what we call recombinant vaccines, or to use this bacteria for the benefit of the human being, like the bacteria that is used in producing yogurt, for example, the zabadi. So, or other dairy products, or other types of crops. So the bacteria has a lot of utilizations in the industry. So that's why we need to know the genetic component or makeup of these bacteria. Other reasons why we culture bacteria in the laboratory is to count their number in a given in a given sample. For example, this is mainly used in the industry. For example, in production of yogurt, in the production of certain type of cheese. They need to count this number of bacteria in a sample of the product, so that they can note the viability or the validity of this product, as if you know they want to define a shelf life اللي هي مدة صلاحية البرودكت. So that's why this is we need to count the number of the bacteria in a sample, or maybe you need to count number of bacteria in a given sample from a patient to know how much is this bacteria causing disease, or how is it very aggressive or virulent in the sample. So the other reasons is we need to, for example, if you have um, a sample that contains a different type of bacteria, different genera, so you need to culture it in solid media so that you can separate each bacteria by itself, because the bacteria, one bacteria can grow and give a colony. So a colony is it indicates one um, one type of bacteria. So if you want to separate different types of bacteria from each other, then you need to culture it on a solid media. And we are going to speak about the different types of media now in the coming part of the session. So if you want to separate it, then you need to culture it in the lab to separate it so that you can study different types of bacteria. So the culturing in the media in the laboratory is very important, and these are part of the reasons why we need to grow bacteria in the laboratory. For the composition of the culture media, of course, as we mentioned before, the culture media should contain all the components required for the bacteria to produce its nutrition and to be able to grow in the required temperature, and the pH of the medium should be adjusted according to the need of the bacteria. But the most important thing that constitute most of the bacteria, bacterial medium, the medium is the where the bacteria grows, providing all the nutritional needs. So definitely it has water, it has source of carbon and energy, and of course to build amino acids and protein, there should be a source of nitrogen. There are trace elements, and there are some growth factors. For certain bacteria might require vitamins, might require certain compounds, that is like sulfur or any other type of phosphorus. So it's according to the bacteria, but most of the types of the medium, they contain these components: water, source of nitrogen, of carbon, source of energy, source of nitrogen, and trace elements, and some growth factors. So these types, these sources, usually produce from normal food that is available for human beings, like meat extracts, yeast extracts, peptone, agar. Agar, it is an extract of certain type of fungi. Of course, there is water, there is casein hydrolysate. So these are normal components or extracts of this to provide a source of nitrogen, source of carbon, source of energy, source of inorganic compounds if the bacteria requires inorganic compounds, and source of trace elements like magnesium, like calcium. Certain bacteria, they require these trace elements. So the culture media itself, it is classified into in a core. Its classification is based on different ways, either based on the consistency, that is it solid, is it liquid, is it semi solid. So it's according to its consistency. The other group is based on nutritional components, And the last category is based on functional use. So, of course, a medium, it could be a solid, but it has certain function. So these are integrated. These ways of defining the culture media are actually integrated. They are not separate. So according to the consistency, if it's a solid medium, it could be has a different function or a specific function. And it could that's why it's because it has a specific function, then it has specific nutrients found in this medium. So based on the consistency, the media are divided into four types. Liquid media, and these are available that usually put in tubes and test tubes and bottles or flask. And these are usually called broths. They shorba yani, shorba. They are nutrient broths and found in liquid in the form of a liquid medium. And usually the bacteria grows in this medium, but they are not growing in the form of colonies. They when they grow, then the medium changes from being clear medium, that is as a new medium, into turbid medium. That means because the bacteria starts to multiply, then the turbidity of the medium increases. That's why this type of medium, liquid medium, is used for the bacteria that if you have a sample that you think it contains very little amount of bacteria, cannot be diagnosed directly from the sample, then you need to grow it into a liquid medium so that it can multiply and increase in number in a faster rate. So, but you have, of course, to provide all the required nutritional requirements to provide the pH, the suitable pH, and the temperature. So, this type of bacteria, you will not be able, of course, to differentiate from the turbidity or the or the or the test tube directly the type of bacteria, because there could be a different type of bacteria growing in the same liquid medium, unlike the solid medium, of course, where each bacteria gives a colony, separate colony. So this type of medium is used to increase the number of bacteria in a sample and to detect the viable bacteria in the liquid medium. The other type of solid medium is of medium is the solid media. This simply you change the consistency of the liquid medium from liquid to a solid by adding what we call in the microbiology laboratory a solidifying agent, and usually we use agar. Agar, it is a polysaccharide that is extracted from red algae من الطحالب الحمراء, and it's of course it is processed and manufactured and usually provided from different companies in form of sheets, very soft sheets, or in the form of powder. And we usually add it to this to the liquid medium in form of 1 to 3%, in the concentration of 1 to 3%, so that the liquid medium solidifies and becomes solid. And this is powered into onto petri dishes, unlike the liquid medium in test tubes and flasks and bottles. So in the solid medium, you have to power it into petri dishes, and usually the solid medium which contains the agar, you need to heat it because the agar can be in a liquid form at temperature 95 degrees centigrade, but as it cools to 40, then it solidifies. So you need to pour it on the petri dish after you heat it. And of course, all the medium, they have to be sterilized, and we are going to speak about the preparation in the coming part. And then then pour it in the petri dishes and use it. The benefit of the solid medium is that you can have different colonies. The bacteria grow in different colonies, so each colony is a growth of a single bacterium. It multiplies and multiplies and gives a colony. So this is the benefit of the solid medium. The third type of bacteria, according to the consistency, is the semi-solid medium, and this is usually contains less concentration of agar, around zero point 0.2 to 0.5%. And usually this type of medium, we use it for testing the motility, if this bacteria is motile or not. That means it contains, if it is motile, then it contains flagellae, or if it is non-motile, so it's a sort of identifying the bacteria from its motility. And this is powered usually in specific in tubes, or in U tubes, to give a room for the bacteria to move. um, and also it is used for to cultivate microaerophilic bacteria. Microaerophilic bacteria, that means the bacteria that that grows in the presence of carbon dioxide. It doesn't need oxygen. So this type of bacteria can be grown in semi-solid medium. So I can show you one of these tubes. So like this tube, it's as you can see here, is a solid, is a semi-solid medium in a tube. And this bacteria is not, it's we inoculate the bacteria in the center, and then it tends to spread all over the medium. That means it is motile. If it remained just where the bacteria is added in the semi-solid, just in the center, that means the bacteria is is uh, is non-motile. It grows where it is just inoculated or cultured. It's the same as this one also. It tends to start to spread, and this shows that even this bacteria requires oxygen it grows much on the surface, and then it spreads according to the center only. So this one is motile, this is non-motile, and this is semi-motile, or it's tending to spread. So this is how we test for motility with the semi-solid medium. So we go back to our session. Is the fourth type of medium is the biphasic media. So this type of biphasic medium, that means it has two phases, solid and liquid. So we use certain type of bottles for this, where we power the solid medium and then we add the liquid medium, and this is usually used in certain type of cultures, especially if the bacteria is very is causing disease, is very infectious or contagious. So just to minimize the opening every time of the bottle and to minimize the contamination of the medium, so we have this biphasic, where the bacteria culture is added to the liquid part, and then it starts to multiply, and then we allow it to grow into the liquid phase by just tilting the bottle, and then the colonies start to grow on the solid phase. And then we can easily take, and if you want to study this bacteria, we can just open the tube or the bottle and take a single colony and then study it. So this is just to minimize the contamination and subculturing of the bacteria. When we say subculture, that means, as you know, from the growth curve of a bacteria, it grows to a certain um, to the from the lock from the lag phase to the lock phase, and then from the lock phase then goes to the stationary phase. When it reaches the stationary phase, you have to subculture it. That means you take it into you grow it into a new medium. That's what we mean by subculture, because we don't want the bacteria to die and to go into the decline phase. if you remember the cross curve. That's why we need to subculture the bacteria every time and then into a new medium so that it will not die because of the depletion of the nutrition in the medium. So these are very important classification according to the consistency. As you can see here, these are the different types of the media. Here, this is the biphasic medium in a bottle, where the solid medium is found in the in a slant form, and the liquid medium is found in the rest of the bottle. So this is what we call solid and liquid medium. The other type of classification is the classification based on the nutritional components, and as I mentioned, very important that the components of the nutrition should fit the type of the bacteria you want to study or you want to culture. According to these nutritional components, there is three types of medium: simple, complex, and synthetic. So most of the nutritional components, basic nutritional components, are found in the simple medium. That means it contains water, it contains nutrient agar, and certain type of nutrients that is required by the bacteria. That is a basic medium, and usually the bacteria that grows in simple medium, we call it non-fastidious bacteria. That means it doesn't require very complicated nutrients for its growth. That means it can grow in very simple medium because it just needs water, it needs a peptone water. This is a meat extract, زي شوربة, meat extract. And that because the meat extract has inorganic salts, it has organic, it has nitrogen source, it has a carbon source. So this is what we call all the types of medium. It has this simple type of medium. So the bacteria that grows in this simple medium, we call it non-fastidious bacteria. For the complex medium, we usually add extra components like blood. We add blood, and we call it blood agar, usually in a solid, in the form of a solid medium. And this because the blood, it contains all the required um, nutrients that is might be needed by all types of bacteria, because the blood, it contains ions like iron, ferrous. It contains proteins, contains carbohydrates. So the normal blood, and it's usually the blood is taken from animal bloods, and is added to the medium. So certain types that of bacteria that grow on such type of media, we call it complex media because there is extra nutrients added by the blood. This type of bacteria, we call it fastidious bacteria. That is means it doesn't grow in a simple medium, but it needs certain type of highly nutritive components. Synthetic or defined media, and this is usually a type of bacteria that is used in the research. So you you um, you develop your medium according to the bacteria you want to study for your research, or you want to study a certain component, nutritive component, its effect on a bacteria. So this is what we call the synthetic bacteria. It is the components of the bacteria. It depends on how you build it up, or the components you build it up by the research requirements according to your research. The third group is classification of this media according to the function, or its use and application. So there are different types of media. There are the basal media, and this is usually replica or similar to what we call simple media. Enriched media, selective or enrichment media. There is a difference between enriched media and selective enrichment media. So don't confuse between them. There is what we call indicator or differential media. There is transport media and anaerobic media. So these types of bacteria are divided according to their application and function and the type of bacteria that will grow on these different media. So the basal media is basically simple media, as I mentioned, that contains water, peptone water, and nutrient agar. So it's a very simple media, and this is used for non-fastidious, as I mentioned. That is a very simple bacteria. It doesn't need much of the nutrients. So this is what we call a basal medium. Usually, the basal media it is found in all types of media, because this is the minimum requirements required by a bacteria. The other type of bacteria of media is the enriched media. So in this type of enriched media, that enriched that means it having extra nutritive components or nutrition components. So, for example, we add blood, serum, or egg yolk to this enriched media, and this will enrich the media. That means it will change it from basal medium into having extra nutritive value. So, unusually, this type of bacteria, this type of media is used for fastidious bacteria. That means that requires very high nutritive value medium to grow. So, such type of medium, we call it, for example, blood agar, chocolate agar. Chocolate agar, it's the difference between blood and chocolate agar. Blood is fresh blood added to the medium. Chocolate agar, it is clotting blood added to the medium. So it is the same components, but blood it is fresh blood. Chocolate it is clotted or coagulated blood added to the medium. So, but at the end, it is enriching the medium by more nutrients, and this where the fastidious bacteria can grow. But in this type of medium, then we can use it also as a differential medium, because in this photo, for example, this is blood, and if this is no there is no growth here in this petri dish because this is a solid medium. But as you can see here, in once the bacteria start to grow, then we can differentiate the different types of bacteria according to their hemolytic effect. That means hemolysis of the red blood cells within the medium. There is certain type of bacteria. There are three types of bacteria can be differentiated from this type of medium, according to their enzymes and according to their ability to hemolyze red blood cells within the medium. There is what we call beta hemolysis. There are certain bacteria that cause complete hemolysis of the red blood cells, and it is called beta hemolysis. There is another type, we call it alpha hemolysis, where there is partial hemolysis of the red blood cells, and they tend to be green in color. They give a greenish color. And there is no hemolysis, what we call gamma hemolysis or gamma bacteria. And this type, there is no hemolysis. So using the blood agar, we can also differentiate the different types of bacteria according to their hemolytic effect. The third type of medium is the selective enrichment medium, and this is we call it selective because it enhances the growth. If you have a sample of a clinical sample from a patient or from an animal or from an environment, soil or whatever, of course it contains different types of bacteria. So using the selective medium, it will inhibit certain type of the growth of certain type of bacteria and enhance the growth of other type of bacteria. That's why we call it selective and enrichment media, because it is selective in the sense that it inhibits certain type of bacteria because it has certain factors that prevent the growth of certain type of bacteria, while it enhances or enrich the growth of others. And this type of bacteria and this type of media, we can use various approaches to be effective by its enrichment, by adding certain type of nutritives, or by changing even the pH of the medium, so that this pH is suitable for certain type of bacteria and enrich its growth while it inhibits other type of bacteria. That's why we call it selective enrichment medium. Example of this, what we call selenite F broth, and this is again could be a liquid, it could be a solid medium, depends on what type of media you want to use. But an example of this is selenite F broth, and this is again it separates pathogenic bacteria that causes the disease from fecal samples. For example, if you take a fecal sample from a patient and you want to detect um, the uh, the pathogenic bacteria in this sample, so you have to you have to grow the bacteria in a medium that prevents the normal bacteria that is found in the body. That means we have two different types of bacteria that could be found in the body. Commensals, that means the normal bacteria that is found in the intestine, in the mouth, in the gut, and so on. But if the patient got sick, so you will take a sample from its fecal sample and grow it into a selective medium to prevent the growth of commensals, that is the normal bacteria, but enhance or enrich the growth of pathogens. So in this case, we can use one of this type of medium is selenite F broth. So we take the sample, it enhances the growth of the pathogens, but at the same time inhibits the commensals. Because of course, to diagnose the disease, you want to look just for the pathogens, the bacteria that causes the disease. So this is type of bacteria we call selective and enrichment. So it has two functions, selective and enrichment. Differential media or indicator media, and this is again, this is type is used for differentiating between different type of bacteria. Like the blood agar, as we mentioned before, blood agar is called differential medium, and but at the same time it's an enriched media. Rich media because it has all the nutritive value. But at the same time, it can be used as differential media because we can differentiate between those cause hemolysis, beta hemolysis, alpha or gamma hemolysis. There is another type of medium also like McConkey agar, and this type of bacteria, for example, it is used to differentiate between the bacteria that found in the intestine, or is it commensal or pathogenic? Because this type of bacteria, this type of media, it contains a sugar which is lactose. So certain type of bacteria, they have the enzyme to hydrolyze lactose, the lactase enzyme to hydrolyze lactose. Others, other type of bacteria found in the gut or the intestine, they don't have this enzyme lactase to hydrolyze lactose. So we can differentiate, as you can see here, if it has this enzyme, then it produces lactic acid, so the medium tend to change into red. While this, because it doesn't hydrolyze the lactose, it doesn't have a lactase enzyme, so it remains as the color of the medium. So this is what we call differential in identifying the type of bacteria according to their enzymes. So it's usually used, this McConkey is used in enterobacteria. We are going to speak about the different types of bacteria, the classification of the bacteria. But this is used in enterobacteria to differentiate lactose fermenters and non-lactose fermenters. So it is differentiating, the same as blood agar. But we are testing here the type of enzymes already found within the bacteria. There is another, the fifth type is the transport media, and this is usually a medium that keeps or preserve the sample. If you are taking a sample from a patient, for example, at home or something like that, and you want to send it to a laboratory for testing, for diagnosing the disease, or further investigation in the laboratory. So you have to take this sample in what we call transport medium. So the transport medium preserve the sample, or the specimen of your before you send it or while you send it to the laboratory. And this type of transport medium is used widely in microbiology laboratories, as you can see here. Prevent drying of the specimen and maintain the pathogen to its, if there is commensals and pathogen, so it will keep the number to the same ratio. They will not, it doesn't, this type of medium doesn't allow the multiplication of the bacteria in the sample. It keep it as its number, so it is preserving the sample. So there are different types of medium, Carrie Blair medium, and other type of medium used. And usually, this type of medium is found in a tube with a swab, where you take the sample with the swab and then put it in the tube and seal it, to prevent any deterioration for the sample. As used, for example, in cholera, if you want to take a diarrhea sample, or even if you want to take a swab from the mouse, or so you need to keep the sample from drying and to keep the pathogen from multiplying further. So this type is very important in the microbiology for processing the samples. The sixth type and the last type of media is the anaerobic media. And from its name, and we already defined before what is anaerobic bacteria, this type of medium, it's a broth medium that contains a source of carbon and nitrogen, usually minced meat, or because, and this is used for growing anaerobic bacteria. So usually as you can see here in the tubes, there is a minced meat in the bottom, and there is a broth or the simple medium on the liquid medium on the top. And usually we expel the oxygen from this type of medium by boiling the medium to expel the oxygen, and then we lock it, and when it cools down, then we add the bacterium. And this is very suitable for anaerobic bacteria like Clostridium, where they grow and in the absence of oxygen. So this is what we call anaerobic medium. And usually we add meat because this type of bacteria, they depend mostly as a source of energy and oxygen oxidizing or chemical processes or anabolic and metabolic processes on the oxygen found in the organic compounds. So they don't utilize oxygen molecules, or oxygen, the oxygen that is found in the air. No, they use the oxygen that is found in the organic compounds. That is why we need to provide as much as possible of the organic compounds in the form of meat so that this bacteria can utilize all its source of carbon, oxygen, and energy from these particular food components. So this is the different types of medium. Now we are going to speak about the preparation and preservation of culture media. For the preparation and preservation of the culture media, of course, these media are all what we call artificial medium, that is prepared from different chemical compounds that is needed by the bacteria. Usually nowadays there are companies and big providers for this media. And usually they are purchased from these companies in the form of powder or what we call dehydrated medium. And you need to prepare it in the lab according to the instructions and guidelines provided by the company or enclosed usually with the pack or the bottle of the medium. So this dehydrated medium, when once you purchase it, you need to. It's very important to store it according to the instruction provided. So usually they are stored in a cool, dry place and protected from sunlight and dust. And beside it, we need to monitor the temperature and humidity of the room because this media is very sensitive to moisture and to temperature. So this is very important before how to store this dehydrated bottle before you prepare it. So if you want to prepare it, then you need to follow 8 steps before until how you use it. So these 8 steps, we are going to define these steps, 8 steps as they are explained here in the diagram. The first step is you need to calculate and weigh the media according to the volume of medium you want. For example, if you want to prepare a solid media and you want to pour it in the petri dish, you need to know how many petri dishes you need for your experiment or your bacteria in the laboratory. So each petri dish might require between 20 to 25 mil. So you need to calculate how much of the dehydrated medium you want to weigh to prepare the volume you want to require. For example, if you want to prepare 10 petri dishes, then definitely you want a volume of 250 mil. So for this 250 mil, you have to read the instructions of the media, how much of grams of the dehydrated medium you want to weigh to prepare this 250 ml. The same thing for if it is a liquid medium. For a liquid medium, usually the test tube takes between 10 to 15 ml. It's according to the size of the test tube and the flask you are using or the bottle you are using. But usually the test tubes they take between 10 to 15 ml. Usually 10 ml is preferred. So how many tubes you want to prepare? So you need to calculate the volume of your solution and then how much grams you want to weigh from the from the dehydrated medium to prepare this volume and then distribute it to the tubes. So the first thing is calculation and weighing. The second step is mixing with water and heating. Of course, you want to dissolve this powder in the water you are using to prepare the medium. So usually, first of all, we have to use distilled deionized water. Distilled deionized water, that means that is you will make sure that this water it doesn't have salts, it doesn't have contaminants, it doesn't have organic compounds like other type of residues in the water. So you need for all types of media you have to use distilled deionized water. So once you add your water to this dehydrated medium, you will mix it with a mixer stirrer in the lab, and then you heat it. You heat it until it dissolves. So this is very important. You have to make sure that the medium is dissolved before you go to the third step. The third step is sterilization اللي هو التعقيم. Sterilization is very important component or process in microbiology laboratory. It's very crucial. So you have to sterilize your tubes before you power the medium. You have to sterilize your petri dishes if they are glass. You have to sterilize them before you use them. So sterilization is very important. We are going to speak about the different processes of sterilization in coming sessions. So sterilization is very important. So you need to sterilize your medium after you have dissolved it in the water, deionized, distilled deionized water. So once you sterilize it, you want to sterilize it, you use what we call an autoclave. Autoclave is similar to the pressure cooker. They قدر الضغط. So you use, but this is specific for microbiology laboratory. So autoclave, you use the autoclave and you put your medium there, and then you have to make sure that this medium could be sterilized using the autoclave, because there are certain types of medium you should not sterilizing using autoclave. There are other methods that will be written in the instructions how to sterilize this medium. Is it using an autoclave, or is it using, for example, filtering? Maybe other medium we can use specific filters to sterilize the medium. But the common thing that we use the autoclave. Just in very few types of medium, we don't use the autoclave because this will spoil the medium. So if you are using the autoclave, you need to put your liquid medium you dissolved in the water, and follow the instructions of the autoclave. Usually, we sterilize the medium at 121 degrees centigrade when the temperature starts to rise to 121 degrees centigrade, and then we leave it for 15 minutes. And after that, um, in this way, we make sure that the medium is sterilized. Of course, there are instructions for autoclave, for different autoclaves. You have to know the instructions of how to use the autoclave in the laboratory, because there are different types of autoclaves according to the instruction, how to open it, how to close it, and what sort of at what time you have to open it. Otherwise, it's very dangerous. So the fourth step is fine tuning, and this in this case, after we sterilize, we need if we want to prepare an enriched medium, adding blood. So because blood usually is sterile, of course, it doesn't have any pathogens or bacteria. So we add it after we sterilize, because you cannot sterilize blood, of course, otherwise it will spoil the medium. So usually for enriched media, um, we add this if it is blood, um, if it is you want to adjust the temperature, the pH, or you want to add antibiotics. It depends on what sort of type of you want to add to the medium after you sterilize it. So this is what we call fine tuning. If you have to adjust the pH according to the need of the bacteria you want to culture on this medium. If you want to make it more alkaline, then you use sodium hydroxide. If you want to make it acidic, because the bacteria lives in acidic pH or medium, then you add HCl. So it depends on the instructions that is provided by the company on the bottle of the medium. And then after that, you leave it to cool down, and then you distribute it. If it's a liquid medium, you distribute it to sterile tubes. If it is a solid, then you distribute it to um, sterile petri dishes. And this is very important, and you have to monitor this step by thermometer, because if it's a solid medium, you don't want it to solidify before it you power it. You have to make sure that the temperature is around it's slightly above the solidification of the agar, which is between 45 to 50 degrees centigrade before it completely solidified, and you will not be able to power it on the petri dish. And then after that, after you distribute it, you seal. If it is a petri dish, you seal it in a plastic bag, a sterile bag. There are specific bags for keeping the petri dishes, and after they solidify, of course, and then you can keep it in the fridge at 2 to 8 degrees centigrade, not the freezer. You have to keep it in the fridge, 2 to 8 degrees centigrade, and this can be kept up to 1 to 2 weeks according to the instruction of the medium provided by the company on the bottle of the medium. There is very important step, what we call quality control. How to check if this medium is it good for using for culturing. There are different labs. They use different protocols in the lab to check for the quality of the medium. One of these methods, for example, they take a petri dish or a tube and, and put it in an incubator. That is mean in a temperature that is suitable for all bacteria they can grow, to detect if this petri dish, without any adding any culture, if there is any bacteria can grow. And this if appeared that after you incubate it in an incubator, that there is appears any colonies in this medium, that means this medium is not sterile. If there is any turbidity in the liquid medium, that means this medium is not good and it's not sterile. So this one type of quality control. Other labs, maybe um, they use infrared, they use dyes to detect in the sterility of the medium, and so on. So there are different protocols, but the most important step is quality control. The last step is the use. When you come to use this, after you take it from the fridge, you have to leave it to reach room temperature, and then you culture it. You should not culture a medium that is you get what that is cool from the refrigerator. You need to come to room temperature before you use it, and you have to make sure there are no water drops or any condensation within the usually cover of the petri dish. If there are any condensation because of the cooling process, then you have to dry it into the an incubator or dehumidifier. Okay. And for maybe 1 day, 24 hours or less than that, until you make sure there is no condensation. Otherwise, the condensation can lead to contamination. So these are the different steps of preparing and preserving the medium. Thank you, and see you in the coming session.